Hi everybody. In this short lecture today, we're going to talk about enzymes. Enzymes are proteins. We've been talking about proteins, one of the macromolecules. And in this instance, enzymes act as catalysts, and what they do is they speed up a reaction. The cool thing about them is that they don't become part of the reaction, they don't become part of the products, and they're not changed, which means they can be used over and over again. Enzymes are specific for a particular type of reaction. That means one enzyme will only work on one subject. They're reusable. We can recognize enzymes because they tend to end in the word ASE. Examples are sucrase, lactase, maltase. Another one is, of course, amylase, which is in the saliva in our mouths where digestion starts. And they work by changing activation energy. Every time a reaction happens where you go from a reactant to a product, a little bit of energy is needed just to kickstart the reaction. You can see in the graph here's the reactants, and then you need a bit of energy, if you like, just to get it over the hump so that the reaction can go to completion. And this amount of energy right here is called the activation energy. Enzymes lower the amount of activation energy that's required. For example, when you want to strike a match, you open the box, you take the match out. Now, if you just hold it, nothing's going to happen. What you need to do is supply a bit of activation energy, so you strike the match along the side of the box. And in this graph, this is just an example of how a reaction will require less energy with an enzyme than without an enzyme. So how do they actually work? Well, enzymes work on particular substances, and we call these substances substrates. The blue blob, if you like, here is the enzyme, and the square yellow rectangle is the substrate. That is what the enzyme is acting on. A good way to think about this is when we did the lab the other day, the lactose is the substrate and the lactase was the enzyme. Now, by looking at this picture, you're probably thinking, oh, you know what? That substrate can fit right in here. And you would be right. That's just the way these things work. They join together because there is a specific gap in which one substrate can only fit. We call this the lock and key effect. Enzyme molecules, which again, remember, are proteins, this is all about proteins, have what's called active sites, and the active site is where the substrate binds to the enzyme. Now, if it cannot get into this active site, then the enzyme is not going to be able to do its job. Certain factors affect enzyme activity, and these are the same factors that affect protein activity because enzymes are proteins. This is what we were doing in the lab the other day. Heat will denature an enzyme. By that we mean it starts to make it unfold, and then it can't act as an enzyme anymore because the shape has changed. It's still a protein, but it just cannot go that one step further and catalyze a reaction. pH will change the enzyme as well, as will salt and acid and base. Other things that affect it are called cofactors or coenzymes. And a cofactor and a coenzyme is just a little something extra that an enzyme sometimes needs in order to make it work. A good example of this is iron. Iron is an element which must be present in hemoglobin. Hemoglobin is a protein that we found in our blood that picks up oxygen and allows it to be carried around the body to the rest of the cells. In this diagram, we can see the structures of proteins. Remember, proteins are characterized by their structure. When you simply have all the amino acids bonding together to form a protein, we call that the primary structure. Whenever that protein starts to fold, it becomes a secondary structure, folds more so, becomes a three-dimensional structure, becomes tertiary structure, until eventually it becomes a very complex protein molecule. Some substances can prevent enzymes from happening. They are, or rather from doing their job, competitive inhibitors, and these are like fake enzymes that really mimic an enzyme and prevent the substrate from joining in. Here's an example right here where you can see the enzyme in blue. A chemical has actually blocked the active site, and then when the substrate comes along, it cannot actually get involved with the enzyme and the reaction is not going to happen. And then we have what's called non-competitive inhibitors. These inhibitors don't actually bind to the active site, but they have an effect on the enzyme and they cause the shape to change. And when the shape changes, it means that the substrate just can't fit anymore. And it's a bit like taking a lock and changing the shape of the lock, and therefore the key doesn't fit. Just like that. So now that you have done a little bit of work on enzymes, it's time to have a look at some of the worksheets. The first thing you're going to look at is POGL. You're just going to work in your groups and get this done. Please read the directions right up here. Answer the questions. This is a particularly interesting piece of reading about poisoning caused by ethylene glycol, which is an antifreeze. Read it, answer the questions. That should take about 10 minutes. When you have finished that, then please go to the environment and enzyme worksheet. This is a fairly short worksheet and it looks at the different factors that affect enzyme activity. And remember, enzymes are proteins, so if it affects enzyme activity, it affects proteins. And these are temperature, pH, enzyme concentration, and substrate concentration. Analyze the graphs, look what's happening. If I had to ask you what temperature do enzymes work best at, you would have a look at the graph and you can see that the peak is about 37 degrees centigrade, which just happens to be body temperature. As for pH, enzymes like it just a little bit above neutral. Whenever you make something too acidic, right down here, or too basic, it's just not going to work. A couple of questions, answer those, do the very best you can. And finally, on day 1 next week, you will have a short quiz on proteins and enzymes. And in order to get you ready for that, you need to fill in the information here on proteins. Go to the PowerPoint that's on my MIDI. This is the PowerPoint that I showed you in class the other day, and simply fill this in. When you've done that, you should be well prepared. Thank you.